Hello, bio students. Today we're going to continue with lesson number 4, DNA replication. In our molecular biology unit, you're going to need your note taker that looks like this for lesson 4. The note taker has 3 pages. We're going to be filling out page 1 and 2 together as you listen to the video lesson. And then the third page, which has a DNA replication fork template, you're going to try to fill out on your own. Okay, let's get started then. All right. Oops, nope. I want to go all the way to the front. All right, so here we are. Molecular biology, lesson number 4, DNA replication. So we're going to start with the central dogma of biology. I told you last time that this was very important and it's something that you have to keep in the back of your head. And we remember that DNA can make copies of itself in a process known as replication. But DNA carries the genetic information and needs to be able to make proteins as well. But DNA cannot leave the nucleus, it is too big. So then it makes a smaller molecule known as RNA. RNA can leave the nucleus and it goes to the ribosomes to make proteins. So the process in which DNA makes RNA is known as transcription, and the process where RNA makes proteins is known as translation, and this is the central dogma of biology. So this diagram that you see here, I'm going to show you this as we go over these 3 processes that are important. So the first process that we're going to study today is DNA replication. So as you can see here, DNA replication happens in the nucleus. And here you see DNA, a double helix, double-stranded molecule, using one of its templates to make RNA, which is a single-stranded smaller molecule. And that process is known as transcription. Notice how RNA can leave the nucleus and it can attach itself or translocate onto a ribosome and can start making proteins with the help of tRNA. And this is a process known as translation. We're going to look at these processes in more detail in the upcoming lessons. So today we're going to focus on process number 1, DNA replication happening in the nucleus. So again, where does it happen? Well, it happens inside the nucleus. And if you want a definition for DNA replication, it's when DNA makes a copy of itself, makes an identical copy of itself inside the nucleus. And when this happens during interphase, remember during the mitosis unit, cell division unit, during the cell cycle, we know that in the interphase of the cell cycle, which is the longest phase of the cell cycle, we have an S phase where DNA makes a copy of itself so that it can then undergo mitosis. So that's when it happens. And why is it known as a semi-conservative process? I will go over that in just a sec. Also know that many enzymes are involved, and the enzymes we will study in detail in just a moment. So DNA replication is known as a semi-conservative process because notice that the blue parental strand, or actually the light blue parental strand, this strand is being used as a template to make the new complementary DNA strand. So semi-conservative means that you're always going to retain one of the parental strands in order to make a complementary strand, even though the final product is always going to be an identical DNA molecule. So in your notes, you can write down that it means to be what does it mean to be semi-conservative? Well, the 2 copies of DNA, each one contain one old or original strand. Here you see it in gray, and one new strand, and that's what you see in purple here. Okay, so you're always going to retain an original strand that you use as template to make a complementary strand that is the newly made strand. So that's what it means to be semi-conservative, keeping an original parental strand. And what are the origins of replication? So these origins can sometimes be referred to as forks or bubbles. And in eukaryotic cells, there could be multiple origins of replication, whereas in prokaryotic cells, there's only one. So these origins of replication is where the double-stranded DNA kind of opens up like a fork or a bubble. So this is going to speed up the replication process. So instead of just having one origin and then the DNA replicating from that one origin, instead, to speed up the process, notice that you can have multiple origins of replication. And these is just going to speed up the rate of DNA replication. So these are the enzymes in DNA replication that you're responsible for knowing, memorizing, and understanding. So there's 4 enzymes, and now, of course, there's more than 4 enzymes, but for purposes of this class, we are going to learn these key 4 enzymes. So the first one is DNA polymerase. It is known as the builder. Then we have helicase. It is known as the unzipper. We have primase. It is known as the initializer. So in other words, it sets a primer where DNA replication needs to start. And ligase, which is known as the gluer. The Amoeba Sisters video that is also linked to this lesson is very helpful in showing you this whole replication process live. So here we see helicase working its magic, the unzipper. So here you see how the enzyme comes in and it unzips the twisted DNA strands. So this area right here is the replication fork. So we see that DNA is no longer twisted here. DNA is separated, and there's other enzymes and proteins that help maintain the 2 DNA strands kind of stay separated that way until copies are made. Then we see primase, or the initializer, here in orange. And primase kind of sets these primers, and these are little segments of RNA, and these are starting points that will signal the following enzyme, known as DNA polymerase, to come in and start adding the nucleotides wherever the primer is set. So it's kind of like over here, start DNA replication here on this side, start DNA replication here on this side. So that's what the enzyme does. It sets the primers. And then we have DNA polymerase, which is the builder, and this is the enzyme that's going to be responsible for adding the nitrogen bases in the new strand. It's actually going to be responsible for adding the whole nucleotide, right, on the other side of DNA in the new complementary strand of DNA. But there's one thing that is important to note about DNA polymerase. It can only add DNA nucleotides in the 5' to 3' direction. So in the previous unit, we learned what it meant to be 5', 3'. That's the direction of the DNA strand in accordance to the numbering of the carbon bases. So here we see that because the parent strand runs in a 3 to 5 direction, that means that DNA polymerase is going to add the new nucleotides in an anti-parallel complementary 5 to 3 direction. So that's something very important to remember. So here we see the new DNA strand forming in a 5 to 3 direction. And if you go up here, you also see the new DNA strand forming in a 5 to 3 direction. Okay. Now, what does it mean to be a leading strand versus a lagging strand? So, if you look at the way helicase is unzipping the DNA strand, okay, notice that this is the direction in which the DNA is opening up. So DNA is opening in this left to right direction. So it's opening up from left to right. So notice how this DNA polymerase is adding the nucleotides in the 5 to 3 direction, so it's adding the nucleotides in the opposite direction than what helicase is opening up the helix. So this DNA polymerase is going in the opposite direction of the replication fork. Okay. Now here, this DNA polymerase, it is opening up in the same direction as the replication fork. So it's going in the same direction as helicase here. You see how they're both looking towards the right. They're both looking in this direction. So because this strand is being formed in the same direction as the direction that helicase is opening up the double helix, then we call this the leading strand, because it's just a continuous strand that doesn't need to stop synthesis. It just keeps going and going and going and going and going in the same direction as helicase is opening up the replication fork. But again, if you notice the lagging strand, this DNA polymerase, since it can only add in a 5 to 3 direction, it is opening in an opposite direction. So this DNA polymerase, as the fork started opening up here, let's say, it started adding in a 5 to 3 direction, and then it went, oh, wait, I'm going the wrong way here, right? Let's keep adding, but now I have to backtrack and I have to start here also in a 5 to 3 direction, and I'm going to copy this segment again in a 5 to 3 direction, and now I need to stop because the fork is opening in the other direction, so now I need to backtrack and start here and keep copying in a 5 to 3 direction. So what ends up happening is that you have these broken pieces or discontinuous strands of DNA being formed. So we call this the lagging strand because the DNA is getting copied in the opposite direction than what the replication fork is being opened. So then these discontinuous fragments in the lagging strand, we call them Okazaki fragments after the scientist who discovered them. Okay. And finally, we have the last enzyme that you're responsible for, which is primase, or the gluer. And this is the enzyme that's responsible for gluing the Okazaki fragments. Because remember that these are discontinuous strands of DNA. So it's responsible for coming in and joining the pieces. So what is the final result of DNA replication? Well, if you start out with one original parent strand, you are going to use those parent strands to make complementary strands, and you're going to end up with 2 identical DNA molecules after one round of replication. But remember that DNA can replicate multiple times, right? So if you take this first generation daughter strand and replicate again, then you're going to end up with another 2 DNA molecules here. And if you take this one and replicate it again, you're going to end up with another 2 identical molecules of DNA. So after 2 rounds of replication, you will end up with 4 identical DNA molecules. So that's what the final result of DNA replication will look like. So take your diagram on the 3rd and last page of your note taker and see if you can use the PowerPoint to label all the different processes and structures that are located within that DNA replicating molecule.